Hello, this is Mrs. Kohler, and we've been talking about genetics, and so far all the things we've been doing, the traits have been either dominant or recessive. Now today what we're going to talk about are a couple of special cases where it's not just straight dominant or recessive. So those are incomplete dominance and codominance. So we're going to start with intermediate inheritance. Intermediate means in the middle. So another word for intermediate inheritance is incomplete dominance. When this happens, the organism shows both alleles, but they kind of get mixed together. And the result is an organism who looks like some kind of a transition halfway between the parents. So the real classic example is with flowers. If you take a red flower and a white flower and cross them with each other in snapdragons, you will end up with pink flowers. So the flower that is a result is a mixture of red and white, which makes pink. But then if you were to breed those pink flowers with each other, you would get half of them would be pink, but about a quarter of them will be red and about a quarter of them will be white. So that's intermediate inheritance. Now the way that works is that both alleles get expressed a little bit. So the one that has one gene for each is the one that's going to look like it's in the middle. So in these flowers, the gene for white is W and the gene for red is R. So if you have a plant that has two white genes, it's going to be white. It's not going to produce any pigment. If you have a plant that has two red genes, or RR, it's going to be red. It's going to produce 2 doses of pigment. But if you have a plant that has a white gene and a red gene, you're going to get pink because it's going to be a mixture of the two. It's producing some pigment, but not a lot. So the one that is heterozygous is the one that looks like it's an intermediate form. This also can happen in humans with our hair. So our hair can be either curly, wavy, or straight. So this young lady here has curly hair. So her genotype is CC. She has 2 genes for curly. This young lady has wavy hair. She has 1 gene for curly and 1 gene for straight. And the result is wavy. And this young lady has straight hair. She has 2 genes for straight, so she has 2 S's. So 2 curly genes is very curly hair, 2 straight genes is very straight hair, but a gene for curly and a gene for straight makes wavy. Now, there's a second kind of inheritance that is similar to intermediate inheritance, but it has some differences. And this is called codominance. And if you think of the word co, it usually means something you're doing together, like Ms. Long is my co-teacher, we teach together. So in codominance, both of the alleles are considered to be dominant. They're both going to show. They're both completely expressed. So if you think about me and Ms. Long, we both have our own personalities and we both say what we think. We both express ourselves completely. But you see them separately from each other. It's not like we're 2 teachers blended into one big teacher. We're 2 different teachers. So this is my dog. This is Tenchi. He's my baby. So if you look at Tenchi's coat, he has what looks like a white coat with orange spots, or red. So he's spotted. So Tenchi has genes for white hair. So Tenchi has a gene for white hair and a gene for red hair. And what happens is some of his hairs are completely red and some of his hairs are completely white. None of the hairs are in the middle. They're either red or they're white, one or the other. So that's what makes that kind of spotting pattern. He'll get like a blotch of red here, a blotch of white there, a blotch of red here, a blotch of white there. It's just random which gene is expressed. That's codominance. So here's some other examples. This horse is displaying codominance because of the spots on his coat. So these spots show you that it's codominant. This flower has blotches of pink and blotches of white. This blotchiness tells you that it's codominant. And we see this with human blood types. We see it in a disease called sickle cell anemia, where a person who carries this just one sickle cell trait makes normal blood cells and blood cells that are shaped wrong. And also the proteins that control our cholesterol are codominant. So I mentioned that codominance occurs in blood types. So there are 3 genes that control blood type. There's an A gene, a B gene, and an O gene. A and B are codominant, and O is recessive. So there's a little table on your notes that says blood types, and the left column says type, and the right column says possible genotypes. So type A, this will be your first column, and these are your possible genotypes. Now, these aren't just AA and AO, it's, the letter I with a capital A. That's the standard symbol. So you can be type A by either having 2 type A genes or a type A gene and a type O gene. You can be type B by having either 2 type B genes or a type B and a type O. Because remember, O is recessive, so that one's not going to show. If you're type AB, you have 1 gene for A and 1 gene for B. And if you're type O, you have 2 little i's. Little I stands for type O. So you should fill that in your table. Now, we're going to do a practice blood type problem, and this is sort of like a paternity test. So, Jane has type O blood, and her husband Jake has type AB blood. I know, my names are very creative. The children that they have type A and type AB blood. So they have 2 kids, one's type A, one's type AB. Jake thinks that Jane has been stepping out on him because he doesn't have type A blood and Jane doesn't have type A blood. So he thinks that he's not the father of this baby. So we want to actually see if he's correct. So the first thing we have to do is figure out each one's genotype. So Jane has type O blood. So her genotype is going to be II. Jake has type AB blood, so he's going to be IAIB. That's Jake's genotype. And then what we have to do with that is we have to make a Punnett square. So let's make the Punnett square and find the possibilities. So we're going to put Jane here, little I, and we're going to put Jake here, IA, IB, and we're going to do the Punnett square and see what we get. So IA, little I, IA, little I, IB, little I, IB, little I. That's what you end up with. So the possible genotypes of the children are IAI and IBI. So this child could be type A, and this child could be type B. Now the question is Jake the father of the type A child? Well, the answer is yes, he could be the father for the type A child. However, he is not the father for the type AB child. Now, chances are, because Jane's type O, she also can't be the mother of a child that's type AB. So maybe the baby got switched to the hospital or something. But Jake could be the father of the type A child, but not the type AB child. So that's what you're going to do when you do blood type problems. See you in class. If you have any questions, talk to Mrs. Kohler or Ms. Long. Have a great day.